think it's 3. It was 3. It was 3. Right, 19 days. GPCR activation, vector molecules, pathways 2. Okay, cool. So you missed the whole thing where we're going over this because I forgot to turn it on, and I've only had one. I had one earlier today, and I had a really good one before, but I guess we're gonna go back to it. Okay, cool. One that I did not record. All right, the GPCR alpha subunit, the G alpha Q activation of phospholipase C. Right. Okay, cool. So this is specifically asking about the one via G alpha Q, the GPCR activation of phospholipase C. Okay, cool. So for this one, imagine with me, if you will, GPCR receiving a signal, the alpha, beta, gamma going to it, except this alpha is different. This alpha is an alpha Q, and instead of attaching to the E protein, which it normally would, this one connects to PIP2, and yes, PIP2. No. I'm sorry, PLC. PLC attaches to PIP2. Which attaches to DAG, but PLC also activates inositol 3-phosphate, which then goes down and it signals to the nucleus to release calcium. No, wait, that wouldn't be the nucleus. It should be the smooth endoplasmic reticulum. Anyway, release calcium, which then resets the whole system. Meanwhile, the PLP2, the DAG binds to the phospholipase C, and that is that part of the pathway. And I've got a couple more. I have the original G protein that we've looked at before, or maybe you didn't see because unfortunately I wasn't streaming properly. And then we have this one. So let's see, DAG, yes, DAG goes to protein kinase, cool. So you can see the whole thing here. And then we have this other one. We have this, so this was the description of the indirect pathways. There's another one, which is with the phospholipid, phospholipase, and this is the direct pathway, and it goes straight to the signaling of a pathway to convert arachidonic acid into cyclooxygenase and epoxygenase, or cytochrome P40, and it goes through these, or lipoxygenase. And I'm not really familiar with these pathways, but maybe we will be in the future. Okay, yes, this is the smooth endoplasmic reticulum. That makes more sense. Okay, cool, moving on. All right, yes, more GPCR proteins. Activation, effector molecules, pathways 4. Cool, yeah, because we've been through 3. Okay, so beta gamma subunits. Okay, cool. So the alpha has been the active subunit that we've been focusing on up until now. Now we see that the beta gamma actually does have a role. It can modulate enzyme activity, protein like transduction transmitter protein. Shoot, I always forget. The proteins, yes, and then a specific example of this is in the muscarinic receptor M2R in the heart. Yes. Beta gamma subunits can also modulate signaling. Yes. Okay. All right. Summary of key concepts. Okay. The net osmotic effect will depend on its ability to permeate the cell membrane. All right. Hormone action at the target tissues. Receptor location and specificity. Physiological action. Receptors for water-soluble, lipid-insoluble hormones are on the cell surface, yes. Receptors for lipid-soluble, water-insoluble hormones are in the intracellular compartment. Now, I would also expect this to be inside of the cell membrane, but it does not say that, because the intracellular compartment is, in fact, an aqueous solution, so it's a little bit funny, but whatever. I understand lipid-soluble, but water-insoluble? All right, amplification of responses. Hormone actions at target cells can be amplified by the successive generation of signaling molecules, intracellular signaling molecules. Then and I think large numbers is in there too. For example, one molecule of a given hormone can lead to the modulation of thousands of target proteins. Yep, see, large numbers of intracellular. All right, cool. So the second messenger system. Cell surface. Yes, receptors for small proteins, peptides, amino acids, and other small molecules, that means calcium, and subdivided into GPCRs with ionotropic and catalytic receptors. And as I was talking about earlier, but you missed it. If we go back, let's see. So cytosolic nuclear receptors are really cool because the nuclear receptors actually change transcription. They operate at fundamental level, where they cause the DNA to actually be transcribed more into more RNA by changing and by modulating transcription factors, which I think is very cool. So moving on. So receptors and signaling 1. Summary of key concepts. Hormones elicit psychological changes, physiological changes by binding onto specific receptors that target cells and tissues. Yes, these receptors occur in different locations depending on the type of the hormone. Receptors for lipid insoluble peptide or blank hormones are on the cell surface plasma membrane. Lipid insoluble peptides or hydrophilic, large, catecholamine. Okay, catecholamines. Cool. Catecholamines, that'd be like acetylcholine on the cell surface. And when we're just talking about the M1 muscarinic receptor, so yeah, so GPCR is a great example of this. Okay, well I can't. All right, skeletal system, major organs. The bones, the muscles, and the tendons. Okay, bones. Oh, right, skeleton. All right, not the musculoskeletal. Sorry, bone, cartilage, bone. I mean, come on. All right, moving forward. We don't need that. All right, ionotropic receptors. Ligand-gated ion channels. General characteristics. Consist of membrane-spanning subunits to associate to form an ion channel. That's right, they do subunits. Like alternating alpha gamma or something. Ligand binding to the extracellular domain results in channel opening and allows cations, sodium or calcium, entry into the target cell. An example is the, okay, can we remember? Ooh, well, an example was, let's see, ligand binding. How about the, oh, I don't know. What? Sodium, potassium antitransporter? Okay, oh, right, yes, okay. So nicotinic acetylcholine receptor. Right, okay, well, oh, well, that makes sense. Yes, we knew that. Yes, okay, so muscarinic receptors are G proteins. And nicotinic receptors are ligand-gated ion channels. And we know that because, like, with pesticide is stimulates. I mean, we know this happens because it increases the chance of. I think it increases the chance of activation potential. It helps to depolarize neurons, if I remember correctly. Wait, I shouldn't mark that as. Even though I kind of knew that, I cannot mark that as done. Okay. Receptors for insoluble. Okay, yes. So catecholamine hormones, cell surface plasma membrane. Yes. So GPCRs as well as ionotropic and ligand-gated ionotropic or ion channels. Yes. So catalytic receptors have an transmembrane domain. I'm not sure. Extracellular domain or domains. Catalytic, okay. Interesting. So they're like catalyzing reactions outside of the cell? Ligand binding to the domains may result in receptor dimerization. Oh, is that kind of it? Okay. Binding of two receptors together to make a functional unit. Doesn't that also happen inside of the... That also happens for transcription factors. Ligand binding to the extracellular domains results in the activation of an intracellular... oh, I have viewers. Intracellular... hello, guys. All right. Ligand binding to the extracellular domains results in the activation of an intracellular catalytic domain. There are 5 major classes of catalytic receptors. Receptor for guanylyl cyclases and blah, 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 blah. You know, that's not really important right now. Okay, cool. We're going to push this forward. That'll be a new card. All right, extracellular. Bam. Ionotropic receptors, ligand-gated ion channels. General characteristics, right? An example is well, we discovered that. That was the nicotinic. And nicotine, it makes it easier for you to think. Or no, it makes it harder for you to think. I think it might hyperpolarize. No, it has to depolarize, right? Because it's an ion channel. It allows the release. Seems like it wouldn't like... So maybe it makes it easier for you to think or for thoughts to come to your head more quickly. I don't entirely understand the neuropsychology of nicotine. Let's see. So, yes. So it binds, the ions now can enter the cell. Ions flow across. So, okay. So if it's allowing ions to enter the cell, like sodium or calcium, then it's definitely going to be hyperpolarizing the nerve cell. Yes, it is hyperpolarizing it. Right. So it relaxes you because you do less thinking. That makes sense. Cool. Yeah, okay, that makes a lot more sense. And this answers a question that I had the other day because we were talking about nicotinoids as pesticides, and where they, I think they literally just turn off insects. Like if you think about it this way, they target the central nervous system, and it's literally an off switch because an overdose prevents them from thinking. Like imagine nicotinoids to be off switches, to be like the class to be an off switch for arthropods. Crazy. Okay. Catalytic receptors, right. Extracellular domains. Not quite explained yet. Okay, ligand binding to the extracellular domains results in activate. Okay, right, intracellular catalytic domain, right, and then guanylyl cyclases. Now, there are catalytic properties of these other receptors, for example, the GPCRs. But maybe it doesn't count. Okay, G protein coupled receptors, GPCR. And why do they all have extracellular domains? Does this have to do with like a common origin? They're all kind of like homologous? I don't know. Let's see. General characteristics. GPCR have seven transmembrane segments. All right. Contain a domain at the 5-6 cytoplasmic loop that binds G proteins. The C-terminal region binds intracellular proteins. There are over 1,000 members of the GPCR family. Diversity of GPCR. Adrenergic receptors, epinephrine, norepinephrine. Let's see, where's more? Muscarinic receptors. Okay. Well, I'm sorry, one viewer, but I really needed some food, so I will talk to you guys later. All right. Peace.